Hello kids, today's video topic is enzyme. So what are the enzymes? Enzymes are biocatalysts who speed up the chemical reaction. Okay, so in our body, so many chemical reaction take place. Enzyme speed up those chemical reaction. Almost all enzymes are protein, but all proteins are not enzyme. There are some exceptions like RNA also act like enzyme. Example is ribozyme. So we know that enzymes are protein. What kind of protein? Protein in tertiary structure act like a enzyme. Okay. So what is the tertiary structure of protein? When the amino acid chain folded upon itself, it produces a 3D structure that is called the tertiary structure of protein. In this structure, you can see there's so many pockets present. So one of the pocket, okay, act like a active site. In this active site, substrate fix and produce the product. So how the enzyme work? See in this picture, this is the enzyme, this is the active site, and this is the substrate. So substrate enter the active site, okay, and the enzyme slightly changes the shape of active site so the substrate can bind to it. So what is formed? Enzyme substrate complex. Then the bonds are broken, new bonds are formed, so new product is produced. So what is formed? Enzyme product complex. Then product leave the active site of enzyme. That's how enzymatic action take place. Now, what is the chemical reaction? In our body, so many bonds are being broken and new bonds are formed during transformation. That is called chemical reaction. So, chemical reaction in the body are called metabolic reaction. Metabolic reactions are two types. Anabolic, where the small substance combine and form the bigger substance, like amino acid form protein. Okay, and second is catabolic reaction. In this, the bigger substance break down and form a small substance, like starch break down and form glucose. Okay, so how we measure the speed of these chemical reaction? By measuring the number of product per unit time, like carbon dioxide and water combine and form carbonic acid. 200 molecules of carbonic acid are produced in per hour. But when there is an enzyme, carbonic anhydrase involved in this reaction, 6 lakh of carbonic acid is produced in per second. So the speed of this reaction is become higher 10 million times. So that's how enzyme speed up the chemical reaction. Chemical reactions are two types. So here is the graph. Suppose that here is the substrate and here is the product. Product energy level is low. So substrate will release the energy and convert into the product. That is called exothermic reaction or spontaneous reaction. Spontaneous reaction. But if product energy level is high, the substrate will gain the energy by heating and convert into the product. Okay, so this is called endothermic reaction or energy requiring reaction. Okay, so the chemical reactions are affected by temperature. So each 10 degrees Celsius change in temperature, whether you increase or decrease. The reaction will be double or half in either direction. Okay. Now next is activation energy. This show how enzymes work. Okay, this is the graph, this is the potential energy, and here is the progress of reaction, the time. So here is the substrate and here is the product. Product energy level is low than substrate, but substrate will not directly change into the product, whether it is exothermic or endothermic. The substrate need to go a little higher energy state to convert into the product. So this is the substrate. Substrate will gain extra energy or free energy and reach to the transition state. Transition state is a unstable state where the substrate is not converted into the product. So what happened here? The substrate gain the extra free energy, okay, and reaches to the little high energy state, which is a transition state. Then it loses that energy which it acquired, and then it loses the energy which it takes here, and then it loses more energy and convert into the product. Okay, so this energy which is required to the substrate to reach at the transition state is called activation energy. When enzymes are introduced, they reduce the amount of activation energy, so substrate convert into the product easily. Okay, so how enzyme works? By reducing the amount of activation energy, so the substrate convert into the product easily. Okay, next is factor affecting enzyme activity. First is pH. Okay, every enzyme work at particular pH. Okay, pH. So that pH is called optimum pH. Like stomach enzyme work at acidic pH. Okay, so if the pH become low to this optimum pH or high to this optimum pH, the enzyme will become inactive and lower rate of reaction will be received. Next is temperature. Enzymes also work at particular temperature that is called optimum temperature. So low temperature to this optimum temperature, the enzyme will become inactive. And when again if enzyme get the optimum temperature, it will become active. Okay. But if the temperature is high to the optimum temperature, the enzyme will denature, because enzyme is made up of protein and protein get damaged at high temperature. But there is some exception. Some enzymes which we get from the bacteria who live in hot condition, who live in thermal vent or hot spring, that enzyme work at high temperature. Okay, that is not affected by the high temperature. Next is substrate concentration. So here is the rate of the reaction and here is the substrate concentration. When you increase the substrate concentration, the rate of the chemical reaction will increase and reaches to a point, okay, that after this, even if you increase the concentration of substrate, the velocity will not change. That is called Vmax. How will you understand? Suppose that here is 10 enzyme If you put 5 substrate, this will give 5 product. 10 enzyme, same amount of enzyme will be same. 8 substrate, how will product you will get? 8. 10 enzyme and 10 substrate, how much product you will get? 10. But here is 10 enzyme, and if you increase the substrate concentration again like 15 substrate, how much product you will get? 10. Why? Because there is no free enzyme is present to bind the extra substrate. Okay, so when we increase the substrate concentration, the rate of the reaction will also increase and reaches to a point that after this, even if you increase the substrate concentration, the velocity will not increase. That is called Vmax, maximum velocity. And half of this velocity is called Vmax by 2. So if you are getting Vmax at 40 substrate, and you will get half of it at 20 substrate. So that is represented by Km. What is the Km? Km is the substrate concentration at which the Vmax by 2 is achieved. Okay, so that is about the substrate, how the substrate concentration affect the enzyme activity. Now enzyme inhibitor. What is enzyme inhibitor? They stop or reduce the enzyme activity. Okay, so there are two types, competitive and non-competitive. First, we talk about competitive. Suppose that this is a enzyme, okay, and this is the active site of enzyme. This is the active site. Here is the substrate. This substrate will fix into this active site and what produce? Product. Okay, now here is a inhibitor. It is inhibitor. It resemble the shape of the substrate. So in place of substrate, okay, the inhibitor will fix into this. Okay, so suppose that this is the enzyme, okay, and if substrate fix into it and produce the product in 5 seconds, okay. But here what happened? In place of this, what fix? Inhibitor. The inhibitor get fixed and it stay here for 3 seconds and then it walk away. Okay. And then what happened? The substrate go and fix into it and produce the product. So what happened? This substrate will take 5 seconds to produce the product. 3 seconds is taken or delayed by the inhibitor. Okay, so what happened? The substrate take 8 seconds to produce the product. So the enzyme activity got delayed due to inhibitor. Okay, so what happened? Example is given in NCERT that malonate is a inhibitor which attaches to the succinic dehydrogenase enzyme and it stop the succinate. What is succinate? Is the substrate to attach to the enzyme. Okay, so that's how the inhibitor delayed the enzymatic action. Now, non-competitive inhibitor. Suppose that here is the enzyme. This is the active site. And this is the substrate. And this is the inhibitor. Here, inhibitor will not fix to the active site. It will fix to any other place. Suppose that here, and changes the shape of the active site. So the substrate cannot fix in active site. So here the inhibitor stop the enzymatic action. Okay, so the topic is done. I hope you like it. Please like, share, subscribe my channel. Thank you.